Hello, my name is Diane Campbell, and I'm excited to be teaching a unit in the Ridge to Reef workshop on environmental data analysis. So I'm going to tell you today about my research on how a changing climate will influence demography and evolution in plant pollinator systems. We know that natural selection plays a central role in the evolution of biodiversity. And for flowering plants, the angiosperms, about 87% of species are pollinated in part by animals, such as this hummingbird, bee, hawk moth, or fly. So selection due to animal pollinators is thought to be critically important to their diversification. We also know that natural selection depends upon the environment, and environmental conditions are changing rapidly around the globe. So this figure shows, for example, changes in average surface temperature from the 1980s to predicted levels at the end of this century. Yet most projections of the biological impacts of climate change do not take into account evolution in response to natural selection. So my research that I'm going to talk about today focuses on three questions. First, how does climate influence natural selection mediated by pollinators? Secondly, how does a changing climate alter the demography and persistence of a plant population? And then third, can a plant population adapt quickly enough to the new environmental conditions to avoid extinction? And this can happen through evolution of traits in response to selection under new conditions, or to plastic expression of traits in direct response to the environment that is adaptive. And this last topic of plasticity is where I'm going to bring in a little bit of material on analyzing multivariate data that will be very relevant to what we're going to do with the data sets for this workshop. So the study system is two species of plants in the genus Ipomopsis, and they're hybrids in the Rocky Mountains, particularly in Colorado, for which we have some long-term data on natural selection and demography. So in this region, the warmer temperatures are leading to drought conditions. And one of the reasons for this is that there's a change in snowpack and thus in the day in the spring that the snow melts over time. So we're seeing progressively earlier snowmelts on average, and those earlier snowmelts mean that plants experience a longer dry period before the summer monsoon rains start in July. And this graph over on the right is showing the snow water equivalent of the snowpack as a difference between now and in the 1950s, and all the red indicates declines. So we're interested in how these new conditions influence natural selection. And I won't talk about all these traits today, but one of the features of this system is that we've already shown that a suite of floral traits influence pollination success. And we've already shown a suite of vegetative traits that influence either growth or survival. And I will talk towards the end about volatile organic compounds that are emitted by flowers that give them fragrances, because this is a multivariate data set that in structure is similar to the one that we'll deal with for the workshop. One of our study areas for these questions is this region in which there's a natural hybrid zone between plants of Ipomopsis aggregata, which occur at elevations near 2,900 meters. populations of Ipomopsis tuba, which occur higher on the mountain at about 3,200 meters in elevation. And in between, there are natural hybrids between these two plant species. So from this region, we have more than 25 years of demographic data and 17 years of data on natural selection on floral traits. And this comes in part from some common gardens that we've planted with more than 9,000 seeds, and from examining more than 500 surrounding natural individuals. And so this all forms the longest running continuous data set on natural selection on the same traits year after year in plants. To explain a little bit where these data come from, the first experiment was started in 1994, in which we established common gardens at different sites. And in each of these gardens, we planted seeds individually using a 10-centimeter grid system, and these seeds had no genetic background. So here's a tagged plant that has emerged at one of our planted locations. We can do this, by the way, because there's no seed bank in this species. And then we would follow the fate of those seeds. The plants spend 2 to 12 years or more as a vegetative rosette of leaves. They then send up a flowering stalk, flower during a single year, set seed, and die. So they have one reproductive bout, they're monocarpic, and when we finally get the seed set, that allows us to estimate the lifetime fitness of the plants, at least through female function. So we have from seed to production of seed. For the data I'm going to show you today, which involves how selection changes over time, I'll show you selection estimates from common gardens at our 2 lower elevation sites where Ipomopsis aggregata normally occurs and where natural hybrids occur. And this involves following what happened to more than 4,600 seeds from 65 different full sib families. All right, so just to show you where these selection estimates come from, they all involve some estimate of fitness. Here, we're looking at the rate at which hummingbird pollinators visit flowers as a function of a trait. And here we're looking at the width of the corolla, which is the collective name for the petals. So plants with wider flowers receive more visits by hummingbird pollinators, and we can get an estimate of selection. It's called a standardized selection differential, which is the covariance between these 2 traits divided by the standard deviation of the trait. So here now you see graphs like this, a measure of fitness, in this case seed production, so this is the lifetime measurement, versus the length of the flower, the corolla length, in different years. And in different years, we see different steepnesses of the relationship, indicating differences in the intensity of selection. So if we take those measurements of selection, so these are the estimates of the standardized selection differential on corolla length, and plot it as a function of the day of snowmelt in the spring for that particular year, you can see that in years with early snowmelt, selection is weak, it's near 0. In years with later snowmelt, wetter conditions, the selection is stronger. And because snowmelt is generally getting earlier over time with climate change, we expect a general weakening of selection with those new conditions. And in fact, that's what we see. If you plot selection intensity versus year, selection is getting weaker and weaker on corolla length. So here's our working hypothesis for what causes this. In wet years with later snowmelt, the greater pollination success of longer flowers can translate into greater seed production. So regardless of whether you have a wet or dry year, the amount of pollen on the stigma is going up with the trait value because those larger flowers are more attractive to pollinators, but only in a wet year can that continue to high levels of seed production. In dry years, it levels off very quickly. And so if you look at plants with this range of trait value, you may not see much difference in fitness. And we're testing that idea with experimental manipulations of snowmelt date in the spring. We can accelerate snowmelt by putting black shade cloths out over the snow in late winter to cause the snow to melt more quickly, and manipulations of summer precipitation using rainout shelters and also additions of water in comparison to control areas. So the second question, how does a changing climate alter the demography of a plant population? We know that with earlier springs, changes in flowering phenology are very common. So here, for example, is a whole community of subalpine plant species. This zero line would indicate a plant species that has not shifted its date of flowering at all. And you can see that for most of the species, they're on the left-hand side, they're flowering earlier than they used to. So those sorts of data are very common, but we have almost no information on whether or how climate change impacts the demography and population growth or decline of particular species. So what happens in Ipomopsis, where we are doing this, is first of all, the number of seeds produced by a plant is lower in years with early snowmelt. So we have seeds per plant as a function of day of snowmelt in the spring, with this dashed line indicating the number of seeds that are needed for replacement of the population. The number of seeds needed to have a finite rate of increase equal to 1. And you can see that in years of early snowmelt, the population drops below replacement. And that's true as well for hybrid populations, also, not just for Ipomopsis aggregata. So in addition to lower seed production, we also see lower seed germination and seedling establishment, lower survival of plants to the next year, and a lower chance of flowering the next year if we get a year of early snowmelt. And we can put all of those vital rates together and how they change with snowmelt into a model, an integral projection model, to look at what would happen to the finite rate of increase for the population. So here's the finite rate of increase versus day of snowmelt. Again, the dashed line at 1 indicating a stable population. This is a population that would be declining, population that's increasing. And if you put together the measured way that all of these vital rates depend upon snowmelt date in the spring, there's a very strong relationship between the predicted finite rate of increase and the day of snowmelt. And these other lines on the graph indicate how much it would change if only one of those vital rates were allowed to depend on snowmelt. And again, we see this for Ipomopsis aggregata. We also see this for hybrid populations. So we can use that information to show that the hybrid population is already below replacement and that early snowmelt is expected to push the Ipomopsis aggregata population below replacement in a few decades. And the way that we've done that is to look at how the day of snowmelt in the spring is changing across years. So this black line here is a regression for that. If you look at the hybrid population from that integral projection model, I'll go back for just a second here. That was hitting a lambda of 1, a finite rate of increase of 1, if the snow melts at about the 140th day of the year. So the 140th day of the year is right there. And that population is expected to be below replacement starting back in year 2000. So it's already below replacement. For Ipomopsis aggregata, that would occur somewhat later, but sometime between 2035 and 2064. So for the third question, can plant populations like this adapt quickly enough to these new environmental conditions to avoid extinction? And this could occur potentially through a process of evolutionary rescue. And this is a cartoon of that process in which a threatened population avoids extinction by adapting to the new environment. So here's the abundance in the population, number of individuals in the population as a function of generation. With a new environmental stress like early snowmelt, the population is expected to decline. But during that process, there would now be natural selection for alleles that are adaptive under the new conditions. And here's how those alleles might change in frequency. As they increase, that would increase the fitness, average fitness in the population, and the population could rebound and start to increase in abundance again. So it would be rescued by evolution. This process has been modeled using quantitative genetic approaches. And we know that in simple models, it depends on the initial growth rate of the population, how much genetic variation there is in a trait, because you can only have an evolutionary response if there's some genetic variation, and how strongly the trait influences fitness. It's also influenced by the evolution of plastic responses to the environment. And models have been extended to include multiple traits and more gradual shifts in the environment. But it's difficult to get all of this information together. And so they have not been parameterized for natural populations. For Ipomopsis, in part because we have more than 25 years of data, including data on genetic variation and heritabilities, we can fully parameterize the models, and that's what we're in the process of doing. So I'm not going to go through the whole thing today. I'll touch briefly on 3 points. How will selection and genetic variance of a vegetative trait like specific leaf area influence the potential for evolutionary rescue? How will that potential be modified by the weakening of selection that we're seeing on traits important to pollination, like the length of the flower? And finally, how plastic are these traits? They respond directly to the environment. And is that plastic response adaptive? Does it increase fitness? So if we zero in on the first part, looking at a functional vegetative trait like specific leaf area. Specific leaf area is the area of a leaf divided by its dry mass. So large values mean thin leaves. Low values mean thick leaves. And we use this environmental challenge of drought due to early snowmelt. And this is just to give some visual representations of that. 2011 was a late snowmelt year. 2012, a very early year. This is almost the same view of some of our study site regions. taken on the same calendar day in those 2 years. Note far more snow here than here. And this is a view again of 2011 versus 2012 on the same calendar day. And note the very low vegetation in that drought year. Okay, so focusing in on this trait of specific leaf area, can selection for that trait rescue the population? All right, so here's some of the parameters for the initial growth rate of the population. Prior to the drought in 2012, the intrinsic rate of increase was hovering around 1, fairly stable population. But in 2012, the mean fitness of the plants was only 56% as high as in the wet year. So it was cut almost in half. We also need to know about genetic variation in the trait. I'll get back to that. And we need to know how the trait influences fitness. So here's how it influences fitness. Here's the probability of surviving to flower the next year as a function of the trait, specific leaf area. And you see this general decline. So selection is directional, favoring plants with low specific leaf area. They have higher survival. And so we've modeled the process of evolutionary rescue with that directional selection. All right, here's that directional selection again, favoring thick leaves, low specific leaf area. And we would predict an evolutionary decrease then in the trait, and so that the change in the trait value between 2 generations can be predicted by the heritability of the trait and the selection differential on it. So we get the selection differential from here. And with that selection, our conclusion is that evolutionary rescue would require very high heritability of the trait. So here's some output from the model: population size versus generation. If you have drought conditions shown in red with no selection going on, that 56% absolute fitness would cause the population to decline towards 0. But if you allow for selection favoring a reduction in specific leaf area, if the heritability is high enough, the population can rebound, and you can see evolutionary rescue. But that's for a heritability of 50%. 50% of the variation is genetic. And if you reduce that even just down to 30%, the dashed blue line, it looks hard to get evolutionary rescue. Although the population starts to rebound, it's going to take many, many generations to get to the replacement level of a stable population, and it's likely to go extinct stochastically before that happens. Our actual estimate of heritability from our common garden studies is that it's around 6%. So that's really low and unlikely to allow this process of evolutionary rescue. So high heritability would be required for that process of evolutionary rescue. And we're currently elaborating on the models using more gradual trends in snowmelt. All right, so if we look at selection on flower traits, how does that play in? Well, I showed you earlier that there can be strong selection on flower length, or at least there was prior to the imposition of early snowmelts. And that can produce rapid evolution because that trait, unlike specific leaf area, has high heritability under field conditions. So we actually predicted how fast it would change over 5 generations. And with the heritability that we measured of 74%, we'd expect this kind of a change. We then actually measured the traits in a whole bunch of populations along an elevational gradient, 25 years apart. So these solid points and solid lines are showing the recent measurements, the dashed ones and open symbols showing the old measurements. And we saw average increases of pretty similar value, 2.4 millimeters compared to the prediction. So prior to drought, this trait was actually evolving. We were seeing selection for longer flowers. The pollinators prefer those. And the black line here shows how we expected population size to change with generation prior to drought conditions. But what happens with imposition of an early snowmelt drought year? Well, now we have, as shown in red, we have reduced absolute fitness. It's only 56% as high. The population would start to decline. And if we add in selection under those drought conditions, it doesn't help. We get pretty much the same decline because selection on flower length is very weak with early snowmelt. The seed production is water limited rather than limited by pollination. So that reduces the potential for evolutionary rescue, and we're now in the process of putting together a multivariate model, including genetic covariances between different kinds of traits. But what I want to spend the last few minutes on is focusing in on this question of plasticity of traits. And I'm going to focus on floral volatile compounds because the data are multivariate. And this is similar to the situation that we'll be analyzing in the workshop, where we're looking at multiple species abundances. So first off, these compounds, volatile organic compounds emitted by flowers and also by leaves, are important because the floral compounds can influence the attraction of pollinators or herbivores on the plant. And we sample these in the field and analyze them with gas chromatography mass spectrometry. So it turns out that Ipomopsis flowers can emit around 50 different compounds. Some of them are shown here. And I'm going to show you a little bit more about two of the compounds, alpha-pinene and indole. Alpha-pinene is the dominant compound produced, and a lot of it, so the mixture, this is showing the proportion of the compound in the mixture, so about 60 to 80% of the mixture is made up of alpha-pinene for Ipomopsis aggregata plants, and almost as much for Ipomopsis tenuituba. Indole is really only emitted by Ipomopsis tenuituba and hybrids, and really not at all by Ipomopsis aggregata. So alpha-pinene, we know, influences seed predation. So oviposition by this fly in the genus Stelia increases with the level of alpha-pinene. Indole, we know, affects visitation by hawk moths and therefore reproductive isolation between the two species of Ipomopsis. And we've done this by using an experiment where we had arrays of inflorescences of 4 types. Red means Ipomopsis aggregata, white means Ipomopsis tenuituba, and the chemical structure above is indole. So half the plants, we added 1 nanogram of indole per flower. And when we did that, if you look at what happened on the Ipomopsis aggregata plants that don't normally produce indole, when we added indole to them, we got 3 times as many hawk moths approaching very close to the inflorescences. So indole attracts hawk moths. The absence of indole means that the hawk moths, at least when they're foraging at nighttime, don't visit this species. Okay, so what I'm going to show you then are some data from an experiment in which we have measured plants at 4 different points in time as there's soil moisture declined. So we simply didn't water the plants over this period. So at day 1, the water content in the soil was around 17%. By the end of the experiment, it was down to 2.5%. So we have 4 different points in time, and we're going to compare the composition of volatile compounds among those 4 groups. So we had 28 different volatile compounds, and what I'm showing you here is called an ordination plot. We'll be making these for the data that we have for the workshop. This particular kind of ordination is called a canonical discriminant analysis. And what it does is define 2 axes here, where this first axis is the combination of volatiles that maximizes the variation among the 4 groups. And so as you go from here to here, that's highly correlated with the amount of alpha farnesene. So groups on this side, like the day 8 to 10 and the day 12 to 13 plants, the drought-stressed ones, are emitting more alpha farnesene. The second canonical function is separating plants along this axis, and you can see that the day 1 plants that have the wettest soil are emitting more benzyl alcohol and 1,3-octadiene compared to the drought-stressed plants. Okay, so this is just one method of ordination. And the way to think about ordination of multivariate data is it can be either constrained. So we did a constrained analysis. And what that means is we were looking for a combination of response variables that best separated some predefined groups. Okay, it's constrained to look for variables that separate day 1 versus day 5 versus day 13 plants. And that's in contrast to an unconstrained analysis, which is looking at combinations of variables that explain the variation among all the data points. So you may be familiar with principal components analysis. That's an unconstrained analysis. And the other axis here is really whether it's a parametric analysis working with Euclidean distances or an analysis that's using some other way of defining differences or dissimilarity between data points. And so in the workshop, we're actually going to use these 2 methods. So instead of a canonical discriminant analysis, we'll use a method called canonical analysis of principal coordinates. We'll talk more about why we're doing that when we get to the actual data. But briefly, it's because the parametric method that I just showed you, you have to have a sample size exceeding the number of response variables, and we don't have that. So just to show you how a canonical analysis of principal coordinates, when these other methods of ordination would look like for this same volatile data I just showed you. Again, these 4 different polygons are representing the 4 different groups. In the day 1 plants here are separated at least partly from the plants at the end of the drought, day 1 emitting lots of benzyl alcohol, day 12 to 13 emitting lots of alpha farnesene. All right, so in terms of the evolutionary rescue models, we've seen that high heritability would be required for evolution of specific leaf area to allow evolutionary rescue in response to sustained extreme droughts. Weakening of selection for flower length and possibly other flower traits with drought is going to reduce the potential for that process to occur. Some traits, I didn't show you the data for specific leaf area, but it is highly plastic, and that plastic is likely adaptive, so that would help with evolutionary rescue. And we know that floral volatiles are plastic in response to drought. So we're still in the process of putting together all the information to parameterize the evolutionary rescue models and see how likely it is to occur in this situation. And a final summary, early snowmelt due to climate change can reduce the intensity of natural selection on floral traits. Early snowmelt is also endangering these subalpine populations, and that's happening despite the absence of easily visible signs. So in an early snowmelt dry year, you don't see reduced flowering. What happens is there's reduced flowering the next year. But nevertheless, early snowmelt is having dramatic effects on these populations. So that raises the question of whether a plant population can adapt quickly enough to the new environmental conditions to avoid extinction. And what we know so far is that high heritabilities, perhaps unrealistically high, would be required for rescue due to changes in leaf traits. And also bad news, weakening of selection on floral traits is reducing the potential for evolutionary rescue. But we're still in the process of answering this question. So stay tuned for the full set of data and results. And I'd just like to thank all these people who have helped over the years in this research. So thanks for your attention, and look forward to doing the R data analysis with you.